cells analysis, the cells were treated with nanomelanin for 50 micrograms per millimeter or 25 micrograms per millimeter. And the percentage of senescent cells were evaluated using the senescent cells chemical safety kit. Now to the third part, which is the results of the experiments. So as you can see here, the first part will consist of two separate graphs. The first one being the penetration of nanomelanin into the cells following incubation. The second one will present the percentage of cell viability in the presence of nanomelanin. So for the first part, we will test it in two different time frames for 24 hours and 72 hours. And this shows that nanomelanin is concentration dependent. For the second graph, as you can see here, even at the highest dose, which is 125 micrograms per millimeter, it doesn't have any significant effect on cell senescence. And this proves that it actually has low cytotoxicity and the cell viability rates is not damaged. And for the second figure, we will be using the cell senescence kit. And the cells with senescent cells will be stained and dyed blue, as you can see with the black arrow on the screen. And now, for each control groups, you can see that the percent of senescent cells are not reduced, and that proves nanomelanin has low cytotoxicity and no effect on cell senescence. For the next part, we will be seeing the cell migration in fibroblasts, and by using scratcher to create a wound. And so we will be observing the process for 72 full hours and using different doses of varying levels. And even at the control group or the highest dose, you can see that the cell migration, which is the bottom column here, it's not affected, and there is still migration and movement in the cells. So this concludes that nanomelanin does not prevent the cell migration in fibroblasts. And for the last part, for the next part, you can see we will be irradiating the cells with different doses of 3 to 10 grays. And as you can see here, the black bar is the control group, and the gray bar is the one with nanomelanin integrated in it. And so this part right here shows that the black bar, when irradiated with each different varying doses, is actually significantly reduced, which is the cell viability. However, as you can see here, when it's integrated with nanomelanin, there is still certain impacts of the irradiation, but it's not nearly as significant, because as you can see here, the cell viability percentage is still very high compared to the original starting point. So this proves that nanomelanin protects the cell viability against irradiation in fibroblasts. And for our last part, we will be using certain functional genes in fibroblasts under irradiation, and we will see the expressions of each of those genes. And as you can see here, for each different genes, there are different levels of performance, which proves that nanomelanin recovers the expression of functional genes in fibroblasts under radiation. Yes, this is the conclusion. And so we can draw three main conclusions from the study. The first one being squid-ink nanomelanin showed biocompatibility with human dermal fibroblasts. The second one being nanomelanin could protect cell viability from X-rays at different tested doses. And the third and final one is nanomelanin could selectively rescue the functional gene expressions in fibroblasts.
May I ask, how you came up with the idea of the squid-ink?
The squid-ink. The thing about melanin is that, as of any research chemical, if we try to extract it using, if we buy it from a manufacturer, it will be very expensive. And what we are students. The investment rates are still very low. We don't have the economic ability to purchase it right from the get-go. So we came and, as mentioned, we saw squid sacks, which are underutilized and also very cheap. And it was a very wealthy source of nanomelanin, so we used it.